Assalamu alaikum everyone. Today I'll present a research paper titled Expression Profiles of Inflammation-Associated MicroRNAs in Periapical Lesions and Human Periodontal Ligament Fibroblasts Inflammation. The study was conducted by Yu et al. And was published in Journal of Endodontics in 2016. Before I start talking about the study itself, briefly understanding the background, apical periodontitis is an inflammatory disease that develops around the tip of the root, usually because of bacterial infection in the root canal. It leads to destruction of periodontal ligament and surrounding bone, and we already know that inflammation is regulated by many molecules, including microRNAs, which are small non-coding RNAs that control gene expression, while several microRNAs have been linked to inflammatory diseases. Their exact role in apical periodontitis was still unclear. That's what motivated the study. The main aim of the study was to identify the expression profiles of inflammation-associated microRNAs in apical periodontitis lesions and compare them with those found in inflamed human periodontal ligament fibroblasts. And they wanted to investigate whether one particular microRNA, as microRNA 155, contributes to inflammation by targeting a protein called SEMA3A. They collected the tissue samples from 10 patients with apical periodontitis who required periradicular surgery after unsuccessful root canal. They also collected 6 healthy periodontal ligament tissues from patients who had teeth extracted for orthodontic reasons, and these served as control group. Along with the tissue samples, they cultured human periodontal ligament fibroblasts in the laboratory and induced inflammation by exposing the cells to LPS, which mimics bacterial infection. They used several laboratory techniques as real-time PCR. It was used to measure the expression of 6 inflammation-related microRNAs. A dual luciphase assay was performed to determine whether microRNA 155 directly binds to the SEMA3A gene, and they also used Western blotting to measure SEMA3A protein levels. ELISA was used to detect the inflammatory cytokines TNF-alpha and interleukin-6, and gene transfection techniques using microRNA 155 mimics and SEMA3A specific to have a better understanding of their functions. And finally, they analyzed all of the data using independent sample t-test, and every experiment was repeated at least 3 times to ensure reliable results. The first major finding was that all 6 microRNAs they investigated were significantly increased in apical periodontitis tissues when compared with healthy tissues. Among them, microRNA 155 showed the greatest increase. However, when they looked at the inflamed periodontal ligament fibroblast, the pattern was completely different. 4 of the microRNAs, which are 29B, 106B, 125B, and 198, were actually decreased, while microRNA 143 and 155 showed no significant change. This tells us that the inflammatory response is actual disease. It's different from what we see in cultured cells. And then they focused on microRNA 155 because it showed the most dramatic increase in the lesion tissues. When they artificially increased the amount of microRNA 155 in the periodontal ligament fibroblasts, the cells produced much higher levels of TNF-alpha and interleukin-6, which are important inflammatory cytokines. In other words, increasing microRNA 155 made the cells more inflammatory, and they also observed changes in the expression of the other microRNAs, suggesting that microRNA 155 acts as a central regulator during inflammation. Another important part of the study was identifying the target of microRNA 155 using the luciferase assay. They confirmed that CIMA3A is a direct target of microRNA 155. When microRNA 155 levels increased, CIMA3A expression decreased. Interestingly, CIMA3A was reduced in apical periodontitis tissue but increased during the early inflammatory response in cultured fibroblasts. When the researcher knocked down CIMA3A, inflammatory cytokines production increased and microRNA 155 expression also increased. This suggests that microRNA 155 and CIMA3A regulate each other through the feedback mechanism that influences inflammation. Based on all these findings, the authors concluded that inflammation-associated microRNAs behave differently in apical periodontitis lesions compared with inflamed periodontal ligament fibroblasts. And more importantly, they identified microRNA 155 as a key regulator of inflammation because it directly suppresses CIMA3A. This means that microRNA 155 could potentially become a therapeutic target for treating apical periodontitis in the future. And the study has some limitations. The sample size was relatively small, including only 10 diseased tissue and 6 healthy controls. Most of the experiments were carried out using cultured cells, so the findings may not fully reflect what happened inside the human body. In addition, the researcher examined only 6 selected microRNAs, meaning there may be other important molecules involved that were not investigated. They also recommended that future in vivo studies are needed before these findings can be translated into clinical practice. To conclude, the study improves our understanding of the molecular mechanisms behind apical periodontitis. It highlights the important role of microRNA 155 in promoting inflammation by suppressing CIMA3A and suggests that targeting this pathway may provide new treatment options for patients with apical periodontitis. And thank you.